Hi, everybody. I'm just going to let you look at me on Zoom, and then I'm going to turn my camera off so that I optimize my bandwidth here. So good to see everybody. Thank you so much. Ariel did not mention, but I am a native of the Pacific Northwest. And so I grew up on Vashon Island over a little ferry ride away here. And so it's always wonderful to come back here to the Northwest and visit my hometown. And I was really grateful that Ariel organized some sunny weather for me. You could have turned the temperature up a little bit, but thank you so much for doing that. And again, great to be here. I've been a faculty at the University of Virginia in Charlottesville, Virginia since 2019. Prior to that, I came from University of Southern California. And but through all this time, when I was at UCLA and when I was at USC and now at UVA, I've been part of something called the Autism Center of Excellence, which used to have a connection here to Seattle. And so we'll maybe have a chance to talk a little bit about that. And what I want to do today is talk about some of the work that we have been doing, just kind of, doing a few examples, not an exhaustive set of things, but where we have looked at the phenomic aspects of autism spectrum disorder, connectomes, and various ways of analyzing connectomic data and looking at connectivity in both terms of function as well as structure. And then some more recent work that we've been doing looking at the genomes of children with who've been diagnosed with autism spectrum disorder and what that may mean. And then hopefully we'll have a little bit of time to have some discussion. Go. On. There we go. One of the things which I find interesting about this particular graph is I got my PhD in 1992, which is where this graph starts, and where they started monitoring children was about 8 years later or so. And that in that time, whereas in the early days, about 1 in 150 children were diagnosed with as being on the autism spectrum disorder, and more recently, it's now about 1 in 36 children. And so the change over that time is really quite dramatic, quite remarkable, and it's like a 312 increase in autism prevalence over the last 2 decades. Probably not unfamiliar to folks here in the Institute, that this is really something which is really quite remarkable in terms of an increase. One of the problems, however, though, is that we really lack some sort of a mechanistically informed, data-driven set of biomarkers which unambiguously diagnose autism spectrum disorder. And so this sort of perpetuates this notion of using behavioral phenotypes as our defining characteristics for what we think autism looks like. Various NIH initiatives have attempted to invert this to try to find biomarkers, which if you have these biomarkers, this defines autism. It hasn't quite taken off yet. And so this is a bit of a challenge for us because there's this status quo of kind of inexact diagnostics, inexact treatments, wasted time and resources, potential biases coming into these various assessments, which may be difficult to remove, and other things that may be affecting things and making it difficult to optimize progress for children and for families. One of the things which is kind of interesting about autism is it's clearly a complex, heterogeneous condition. The age, diagnostics, diagnostically, in terms of diagnostics, it can be identified as early as 18 months old. There is an interesting sex ratio with about 4 to 1 boys to girls in terms of diagnostics. There is a very complex and uncertain neurobiological etiology, probably polygenic, very likely heritable, covers a wide spectrum, everything from children who really cannot seem to be able to function where in some cases, whereas in other cases you get children who are very verbal, hyper intelligent, but have very difficult interpersonal interactions. A number of socioeconomic factors are involved, and neuroimaging neuroimaging studies show altered patterns of brain connectivity, et cetera, which we'll get into a little bit. One of the things which was kind of interesting is back in 1992, or right before that, when I got my PhD, this was kind of the model for how autism was being investigated. And sorry, I'm just going to remove this key so I don't bonk into it. Where Uta Frith and others had posited that there was this kind of cognitive dimension with a missing ability that was sort of an area of active research that was sandwiched between the biological and the behavioral and the social. And that this was something that they pursued, looking at how autism, people with autism communicate. And they drew from something called the Logogen model, which actually had its etiology a number of years before, where you had this kind of method of cognitive modeling, which was very popular around the time I was going to graduate school. And invariably, there was some sort of cloud out here that said attention and a bunch of arrows. And if you didn't like where the arrows connected, you could erase the arrows and draw new arrows. And any one of these boxes could be broken out into any number of other boxes as well, to kind of make the model more complicated. And using a model like this and others, a lot of the early work, especially around the time that I was doing my PhD at the University of London, was looking at the notion of the theory of mind. So I'm old enough to remember when the theory of mind idea was brand new, and kind of got its start largely out of the University of London, where I was a graduate student. And the idea was that there would people who had been diagnosed with autism did not have a theory of mind, whereas typically developing children did have a theory of mind. A theory of mind is your ability to put yourself in the shoes of someone else and understand kind of their cognitive reasoning, and that children with autism do not seem to have this ability, according to them at the time. And one example of this was people with autism not being able to perform well on this Sally-Ann task, which is very classic kind of early test of autism, where you have Sally and you have Ann. They have a basket and a box, and Sally has a marble. She puts it into her basket. Sally goes out for a walk, and Ann, being sneaky, goes and takes the marble out of her basket and puts it into the box. And Sally comes back, and where's she going to try to look for her marble? And children with autism did not seem to be able to do well at understanding that the marble's no longer in the basket, it's in the box, and they could not understand where she was necessarily supposed to go. When we would have seminars on this at the University of London, there was a lot of talk about how the children with autism do not have this theory of mind at all. And I remember being in a particular seminar raising my hand and asking, well, is there in between, right? Can you have a little bit of theory of mind or some sort of middle ground of theory of mind? And I was told, no, you can't do that. It's an all or nothing kind of a thing. And I said, well, what about the brain? What makes you think you can create these complicated models of, cognitive processing of the theory of mind without reference to brain imaging or understanding if there's any brain pathology or differences in brain development? And I was told, Jack, we don't need to know about the brain in order to make our cognitive theories. And they were very, very indignant about that. And I always kind of wondered, how can this kind of all or nothing kind of idea, without any reference to brain form, function, connectivity, or underlying disorder, how could it actually be an explanatory paradigm at all? And so it was something I was always very curious about. And so you're all familiar now, of course, that brain research is really complicated. It's data rich. We don't collect less data in human neuroimaging and neuroscience studies generally. We keep collecting more data, very data intensive. And it shouldn't be surprising, because the brain is impossibly complex with, trillions of synapses, and maybe it's estimated to store between, 50 terabytes and 2 petabytes of information. And that neuroimaging data analytics is complex and big, and that able to understand the brain in terms of its form, function, and connectivity, we need very complicated methods to be able to do that. And in addition to all of the neuroimaging data, you also have genome sequencing, you have deep phenotyping, you have other exotica like microbiomes and all that. Is that now we really see neuroscience as being a data science, and that's almost unescapable. And that was one of the conclusions that I came to back in 1992, that without reference to some sort of data, that something like the theory of mind in autism was going to be very difficult to study. And I didn't look at it again for 20 years. And so it's interesting that I've kind of somehow stumbled my way back into it. And I've done so by being part of something called the Autism Center of Excellence. This involves a multi-institutional effort from around the country. It used to involve folks at the Seattle Children's here at the University of Washington, and we are now in our third wave of data collection. So we've had NIH funding now for over 12 years, and we're into our third wave of data collection. Seattle used to be, oops. Sorry, I'm getting a message saying the meeting's going to end in 10 minutes. I hope that's not true. Should I say okay? Okay. And though in our third wave, our Seattle friends are no longer participating in this, we still love you, and we want you to know we miss you terribly. We'll hope that we'll find other ways to interact. But our Autism Center of Excellence Network is a NIMH funded activity that we've done for over 12 years. During this time, I used to be at UCLA over here and at USC, and then I moved over here to the East Coast, and now the whole project is based at the University of Virginia in Charlottesville. And really the whole goal of this is to try to understand and integrate genetics, brain imaging, and clinical phenotyping, in particular to understand the differences between boys and girls with autism spectrum disorder. And increasingly under our new ACE, we are looking at everything in between as well. So gender identity as well. Some of the work I'm going to talk about today is largely looking at boys and girls, but in the future, this is going to be looking at other dimensions of gender identity as well. One of the things that we have done initially is to look at some of the behavioral and phenotypic measures. We actually have a very, very rich collection of phenotypic measures, looking at the RBSR, the brief, maybe many of you are familiar with this, the CBCL. These are all different metrics of cognitive and behavioral functioning, typically given to children with autism spectrum disorder. And this is some work done by Zachary Jacokes. Maybe you might know Wendy Stone, who is Zachary's mom. Zachary is my PhD student, and so he has himself a local connection here at the University of Washington. And this was some work that he did a couple of years ago, where he did a multivariate discriminant analysis of all of these different metrics that we included. These are the ones that were part of the linear discriminant analysis, which showed the biggest results. I'll get to that in a second. He was able to extract two multivariate dimensions from these data. One that clearly separates the children with autism spectrum disorder, which is located on the left over here, with the folks who are typically developing children, which is located on the right, and the purple and the kind of light blue over here. But then there's a second dimension as well, which clearly showed that there is a separation between the boys with autism spectrum disorder and the girls with autism spectrum disorder, but that separation did not exist amongst the typically developing children. So there is something about these metrics, which is not only picking up the difference between those children with ASD and those without, who are typically developing, but also amongst the ASD sample, you're able to separate both males and females. So this is like basically a sex by diagnostic interaction effectively. And going and looking at these, this plot is a linear combination of all of these subscales and all of these subscales and all of these subscales, which produce this effect. So we're clearly able to see that there's something about these particular subjects which is separable based upon these major kind of assessment instruments. One of the things that Zach is going into now, however, is looking, though, at whether or not there are any particular biases that have been kind of baked into these particular metrics over the years. One of the things which is kind of interesting to know about autism spectrum disorder is that back in the day, it was always considered that girls don't get autism spectrum disorder, boys do. And so a lot of the early assessments were normalized on male subjects, not on females. That was kind of more recent. There could be some residual effects to that in some of these, which is something that Zach is beginning to explore, and about how whether or not when we control for what we consider as an inclusion or an exclusion criteria to be in a study like this, whether or not that itself produces a bias, something called a collider bias, which is a whole other talk, but it's a very interesting area. Another thing that we have looked at is trying to understand ASD and the social brain. Clearly, children with autism spectrum disorder have difficulties with social interactions and understanding social cues. One of the things we have done is to do a number of studies that looked at the effects of what appear to be biological motion. Biological motion looks a little like this on the left-hand side, this coherent biological motion, where you have a series of dots which move in some sort of a human-like fashion, dancing around and doing a little, waving arms and that kind of thing. Looks like coherent, biologically inspired motion. In contrast, on the right, you have a bunch of dots which are moving kind of in a scrambled biological pattern. So the same dots, they're just spatial locations, are now moving around in a scrambled sort of a fashion, but it does not give any hints that that might be biological. Looking at this, it was kind of clear that there were 3 different states of how people kind of perceived these particular stimuli. Either there was a kind of state-related activity where the state of having ASD characterized the nature of this potential disruption in brain circuitry, trait activity reflective of shared areas of dysfunction between unaffected siblings and the children with ASD, which may have provided a very interesting neuroendophenotype to facilitate looking at genomics of this, and a compensatory activity, which is unique to the unaffected siblings, suggesting neural mechanisms which might compensate for increased genetic risk for developing ASD. And in looking at this, and going and looking at these children who fell into these particular states, it was possible to go in and do some brain mapping here, looking, for example, where typically developing controls had greater activity than ASD, and that unaffected siblings were greater than ASD, and coloring that as red. Areas of trait activity where typically developing children were greater than ASD and typically developing children were greater than unaffected siblings, and compensatory activity where unaffected siblings were greater than ASD and unaffected siblings were greater than TD, or typically developing children. And plotting those as particular maps, indicating a number of different areas in kind of areas of the kind of inferior temporal regions, areas of possibly hippocampal formation, as well as areas in frontal cortex as well. So we've, again, tried to play with this a little bit more in looking at whether or not ASD females showed markedly reduced responses versus typically developing females, and found that particularly in sensory motor, striatal and frontal regions, this was definitely true. And this did not resemble the differences between ASD males and typically developing males, even though the neural responses did not significantly differ. So this can be kind of seen in these various maps here. We're looking at permutation-based, this is on the left, permutation-based inference, as well as a cluster-wise inference, where looking at the various patterns here. And it's out of this study, which was done by Allison Jack and colleagues based at George Mason University, is that the results suggested that striatal genetic impacts may contribute to the pathways which are at risk, specifically in ASD females. So there could be some specific differences between ASD boys, ASD girls, and typically developing controls. Done a few other things using this exact same task paradigm to look at whether or not artificial intelligence and machine learning methodologies might also be helpful in extracting patterns that help differentiate boys from girls and ASD from non-ASD children. This is an example of looking at a pre-time point and a post-time point relative to kind of some neuroprediction of treatment response in autism children, and showing this is the social responsive score here, looking at their total responsive score, that things changed after they had this kind of treatment response. Looking at biological motion versus the scrambled motion, and looking at these circuits that support social information processing, again, particularly involving the superior temporal sulcus, fusiform gyrus, amygdala, parietal cortex, and a number of other places which are associated with social reward mechanisms. And so it's also kind of interesting that one can build a predictor here, which shows you a predicted change in severity that actually does a pretty good job of predicting the actual change in severity with a very strong correlation coefficient. A lot of these areas which were identified in the brain tend to cluster around some of those same areas that we saw in that prior slide. So we're encouraged by that. Looking at brain morphometry is another dimension. So those prior ones were looking at brain function and looking at a particular task stimuli, that's a biological motion. This is looking at brain morphometry and asking similar questions about whether or not we can use multiple metrics. We talked about function before and trying to look at functional differences between children with autism spectrum disorder and those who are typically developing, and boys and girls in particular. So we've also looked at the structural differences. So taking the anatomical data and making measurements at every different location on the cortex of the cortical volume, of thickness, of something called the Gaussian curvature. So whether something's going like this or something's basically concave or convex. A thing called a gyrification index, giving you an idea of the depth of various sulci, as well as the degree of connectivity as determined using diffusion weighted imaging, and stuck all that into a multidimensional scaling methodology, and also using support vector machines to be able to ask whether or not we can tell the difference between boys and girls with and without autism. And accordingly, we can. And areas, for example, such as the left anterior insula, the right inferior frontal gyrus, the gyrus rectus, and the calcarine sulcus also look like they are areas where there are some structural differences which differentiate between those particular populations. Some of these areas overlap with some of those functional areas we described before, and some of them do not. So kind of interesting, and this is work that was done by my colleague Andre Irimia and as part of our GENDAR research consortium. We've also looked at other things, looking at the connectomes of males and females with and without ASD, and identified that one particular difference they have is in terms of their white matter tractography density, and where we find a sex by diagnosis interaction. All these different areas are differentiated in terms of the white matter connectivity as measured using diffusion weighted imaging. It was at least 64 direction diffusion weighted imaging, where we performed tractography, and we then looked at the density and a number of other metrics related to our subjects. And in looking at these different kind of shades of red and yellow here, you can see these areas which contribute to that sex by diagnosis interaction. The things that are the more yellow are the greatest differentiators between the two. So not only in terms of function, but also in terms of structure and in terms of connectivity, structural connectivity, we're able to tell the difference using some kind of advanced machine learning kind of methodologies, which is kind of neat. Here we go. One of the other things that we've been doing with regard to connectivity is looking at something called the G ratio, which is from diffusion weighted imaging. One can make measurements of this G ratio, but actually the G ratio itself actually comes from a long time ago, and it really is the measure of a particular axon, being able to make a measurement of from the center of that axon to the edge of the lipid bilayer, and a measure of from the center to the edge of the myelinated portion. And if you can consider the inner diameter as little d and the outer diameter as big D, you get the ratio of the two is called the G ratio. And explorations of this, people have explored this G ratio as a way to, if you do a particular transform, you can actually transform it to get a value which is proportional to the conduction velocity of electrical signals through that little section of the neuron, which is pretty cool. This actually comes from a guy named Rushton, who was a contemporary of Hodgkin and Huxley, and actually worked with them to develop this relationship, where if you take this G ratio and you know something about the outer diameter, that with a slight transform, you can produce a value of theta in this case, which is proportional to the conduction velocity. At the same time, that same relationship is also proportional to the internodal distance, where the nodes are the nodes of Ranvier. And so by looking at this, it's possible to find where this particular relationship peaks when you're looking at as G varies. You can see that it peaks at a value of about 0.606, which is 1 over the square root of E. All of this comes out of electrical current theory, which is a whole other lecture. But the idea being that if you have this relationship, and you can make these measurements of this outer diameter as well as this G ratio, you can get estimates of the conduction velocity in any particular location. It's possible to go through using neuroimaging to be able to make these estimates, and we have done so. This is work of Benjamin Newman working in my group. We have a paper submitted to PLOS One describing this work right now, where we can go and get actual axonal volume fractions in every location in white matter. This is just an example of looking at the corpus callosum in typically developing children, as well as autism spectrum disorder, looking at this aggregate G ratio over every point in the brain, and you get something which looks a lot like a fractional anisotropy map, if you're familiar with that, but this actually is more related to the conduction velocity. And then by looking at these in terms of looking at the proportions of extracellular water from a microstructural standpoint, G ratio and the conduction velocity in all cortical ROIs, for example, or in just all the white matter, one can see subtle differences between children who are typically developing and those who have autism spectrum disorder. So you'll see these slight little conduction velocity differences. This is suggestive of that the connections are there. They are just not optimized in terms of conduction velocity. This could be due to 2 reasons. Either those particular processes are the inner diameter are of a different degree of thickness, or the myelination is changed, and so they're not as myelinated as they could be. One of the things you notice about the graph like this is that if this is your G ratio of that degree of myelination, or the degree of the axonal diameter to the myelinated diameter, that if it gets too large, that ratio gets too large, that means basically you have thinner myelin relative to the axonal diameter. The speed of conduction falls off quite precipitously. If you have the other effect, you have too much myelin and that ratio gets too small, you also reduce your conduction velocity. And so something about the brain has optimized it to be able to work at this value of 0.606. But in certain diseases or certain conditions like Alzheimer's or Parkinson's, we're applying this in both cases. We're seeing differences here. There appear to also be velocity-related differences in autism spectrum disorder, which differentiate autism with typically developing children, and we're also looking at the sex differences here as well. And here's just some more stuff looking at proportions of extracellular water, looking at this aggregate G ratio, but then looking at this conduction velocity, where you're going to see that as you're looking at things like social communication questionnaire totals, you're seeing that there's just a general decline in the conduction velocity for all of these gold-colored dots, which are the children with autism spectrum disorder, relative to the blue dots, which are the typically developing children. Now, this is really kind of interesting stuff. We're really excited about this, and so we're really pursuing this hard. So there'll be more on that once this article in PLOS One gets published, we hope. One of the other things that we are doing, kind of the third leg of this particular stool, is looking at the neurogenetics associated with autism spectrum disorder. It's clearly very mysterious, and so having some sort of sense of what that may involve is very helpful. This is some work of Allison Jack and colleagues, which was published in Brain from a few years ago, looking at the regions that were represented in the brainspan developmental transcriptome. This is looking at 10 weeks to about 2 years, and identifying these particular regions in medial frontal cortex, area V1, kind of midbrain areas, and a number of cortical regions, and asking whether or not any of our ASD females have neurofunctional candidate gene sets which overlap with any of those areas. And there does appear to be overlap between dorsolateral prefrontal cortex, primary motor cortex, primary somatosensory cortex, superior temporal cortex striatum, and ventrolateral prefrontal cortex. And so this is all looking at whether or not there are rare copy number variants containing genes which are expressed in these ASD female profile ROIs. And here's kind of a breakdown of this where we're looking at whether or not the ASD females have a greater number of copy number variants than the ASD males. And in each of these different areas there, that appears to be true, suggesting that there is some sort of a divergent molecular pathway involved in ASD boys relative to ASD girls related to copy number variants across these different gene sets. One of the things that should be kind of particular interest is that there's a potential for autism to be an X-linked genetic disorder. And we've started to look at this a lot more closely, whereas the previous stuff is just like looking at, like, for example, kind of risk assessment scores and whatnot. This is looking a little bit more specifically. There is a literature which is growing, which suggests that this may be true. You have spectrums of autism associated with polygenic risk and sex, where they've hypothesized that there's a higher prevalence of ASD in males compared to females, might be due to differences in how the autosomes are affecting each other, but also how the sex chromosomes express themselves differently. And likewise, that there are rare maternal inherited coding variants on the X chromosome, which may carry a particular risk. And so we've been inspired by this and started to look at this more carefully. This is some work which is done by Ian Eteremos, working with me. He's also a post-bac fellow at the National Institutes of Mental Health, and he's been doing some of this work looking at the entire collection of Safari genes. This is the Simons database, Simons Foundation database for autism spectrum, and every one of these little red indicators up here, red shorthand, is one of the genes which is listed in their particular database. This is looking at this kind of a map of every chromosome, and this little gray kind of varying line here is a measure of the mutation density as taken from our particular autism Center of Excellence cohort. And so you can kind of see that you have this varying amount of mutation density that happens to surround some of these areas where these particular genes are indicated. So we've been particularly interested in that. But in some of our analyses, we've started to see this kind of thing that suggested to us that there may be something going on specific with the X chromosome, the sex chromosomes in general. And our hypothesis is that there are structural variants within or near an area of the sex chromosomes, which is called the Pseudoautosomal region, that may play a particularly interesting role in the differentiation between boys and girls with and without autism spectrum disorder. And so we've decided to kind of look at this in some detail. There's really several main research questions. So really, starting generally, which genes, more broadly speaking, are differentially expressed between autistic males and neurotypical individuals when gender and diagnosis are considered as interaction effects, and then which genes harbor a significant number of pathogenic structural variants, in this case, copy number variants. And so our data sets, again, come from the Autism Center of Excellence. I'll describe a little bit about that in a second, but we have, in particular, variant annotated whole genome sequencing and bulk RNA-seq related measures of gene expression. Here's a little bit more about how our genetic analyses were done. You know, pretty standard stuff with Illumina chips, et cetera, and looking at copy number variants. And in particular, our whole genome sequencing was done by the New York Genome Center, and they just do a whole slew of things to give us a very rich annotated set of data. So the first thing that we did was just identifying differentially expressed genes. And so when we do this, this is kind of a description of our pipeline for doing this. We use something called Callisto, and then looked at a differential expression analysis and then a functional enrichment analysis. And the thing that was interesting is that when we went and looked at the top differentially expressed genes, they all show up on the sex chromosome. So you have this one that's showing up on the Y chromosome, and all of these which are showing up on the X chromosomes. And so we're very intrigued by this, because when we go and we look at these are all these kind of interesting protein kinase, Y-linked kind of things, things that are, again, X-linked themselves, other things which seem to have some sort of hint that they may be kind of interesting to look at and located in a particularly interesting region of the genome, which we'll get to in a second. Going and taking these apart and looking what is upregulated and downregulated respectively, we see that this one particular gene on the X chromosome is upregulated, excuse me, and then all of these other ones on the right side of the panel here are all downregulated, and all of these are potentially interesting genes located in this area of the sex chromosomes, which we'll get to here in a second. So we wanted to then, as part 2 of this, identify the genes which harbor these pathogenic structural variants in the X chromosome in particular. And so we took our variant annotated ACE whole genome sequencing data sets to identify the genes that have a high number of mutations in the X chromosome and see whether these genes have a high number of pathogenic variants as well. And so really our research questions are which genes are on the X chromosome and harbor these structural variants, what brain areas might they be associated with, et cetera. And so here's again our kind of little workflow. We've got our annotated whole genome sequencing from the New York Genome Center. We do a digital karyotyping, we perform functional enrichment analysis, and utilize a variety of approaches to establish these particular genes. So when we do this, we find a number, again, there's the top 25 X chromosome genes with the highest number of structural variants, that is deletions of various base pairs out of them. So here's this big kind of pool of them. In going and look at these more specifically, we identify, and looking at their location along the chromosome, the X chromosome in particular, we notice this little clump down here at the left edge. This is this area which is within or around this area called the pseudoautosomal region of the X chromosomes. Here are those particular genes which are in the circle. All of these genes are located either within that pseudoautosomal region or next to it, which is particularly important. So the pseudoautosomal region is this really interesting and weird part of the X chromosomes, sex chromosomes in general, just at the tip here of each of PAR1, which is the tip of the X chromosome, and PAR1 is also located at the tip of the Y chromosome. There's a PAR2, which is located at the end of the long arm, and a PAR2 at the end of the long arm of the Y chromosome. There are about 29 or 30 genes which are located in these regions. They actually go through a pairing during meiosis. So unlike other genes within the X chromosomes that don't do any switching, they do have in the pseudoautosomal region, they do recombinations. And so they're really kind of interesting because you're going to have all of these genes that we've identified in that one end of things are maybe suspects here, and maybe those that are undergoing some unusual combination and maybe something that we want to look at a little more closely. And it turns out that a number of these genes have significant expression in a variety of different brain regions. If we consult the Human Protein Atlas and look up these genes, we can see that a lot of these have associations with various portions of brain development, with brain structure located in areas of the midbrain, in white matter, which may underlie some of those connectivity differences that we've saw earlier. As well as in the cortex and cerebellum. A couple of genes in particular, the SHOX gene is one that's associated with kind of a growth factor, and it's also been associated with autism spectrum disorder, as is this PRK gene, which has both an X and a Y component, and has been identified as being something which is associated with ASD, apraxia, and insensitivity to pain. And these are, two very, fairly recent studies which have associated these. So these areas in the pseudoautosomal region or nearby it may be something that's or suitable for some useful further genetic studies. In looking at doing a functional enrichment analysis, of all of these genes from the pseudoautosomal region, we see that we find that intellectual disability, monosomy, just the presence of one gene in particular, as well as Turner syndrome, which is often kind of related to autism in many ways, are the top most indexed structural variants that we particularly see. So we're kind of excited by that. Going a little bit further and asking whether or not these have any sort of relation to synaptic changes by mapping these into the SynGO functional enrichment analysis. This is where things kind of break down. We just don't have enough statistical power to really kind of say for certain whether or not these have any sort of synaptic or presynaptic, or whether or not these are affecting neurons in particular in terms of their development or whatnot. But we have these little, it hints that there's something going on, but it may not quite be significant. We're not quite powered enough to be able to tell the difference just yet. But it suggests that maybe this is something further we need to do. So we have a number of further steps that we need to take in order to kind of make sure that these genomic analyses are done with a little more fidelity and a little more rigor. So these are just some examples of the steps that we need to pursue in order to make that real. I'm kind of getting towards the end here with really thinking that the future of ASD research is really going to be a data-driven neuroscience kind of endeavor. And I'm kind of pleased because a lot of the people who really went all in on the theory of mind back in 1992 are some of those same people who are really kind of, it's dawned on them that autism research is a big data science, and that neuroimaging and genetics and deep phenotyping are the kinds of things that we're going to need to be able to do and do well in order to make some headway in understanding the heterogeneity of autism spectrum disorder and leveraging these large genomic databases, human neuroimaging, but also how these particular areas of big science and large-scale data analytics are going to affect how we understand the social psychology of autism and the cognitive neuroscience of autism. So pretty pleased to kind of see those same people kind of come around to the same realization that I had back in the day. They eventually got there. So just to reach some conclusions, kind of summarize this for you. Phenotypically, our kind of multivariate analyses that we've been doing really show that ASD boys and girls can be differentiated from each other and can be differentiated from typically developing boys and girls who themselves do not differ from each other. There's something very interesting going on there behaviorally. fMRI results show really notable differences when observing biological versus random patterns of motion. And that has been something that we've been very excited about. And then applying machine learning, and increasingly it'll be deep learning and neural networks and everything else. We'll also be able to explore that data further to separate ASD from typically developing children, as well as from boys from girls. And there are apparent alterations in not just the structural connectivity of the brain, but how that structure begets the function that underlies some of the function and structural connectome, which we think is pretty interesting. And again, we have a paper coming out on that hopefully very soon. These genetic differences exist in terms of total copy number variants associated with a number of different brain regions, but also there's these intriguing clusters of specific genes which happen to be sex-linked around an area of the X chromosome and also the Y chromosome that appear to undergo a lot of recombination during meiosis. And if that process is interrupted or those areas are damaged in that process, it could give some rise to, and an explanation to why ASD is so heterogeneous, why its pattern of inheritance is kind of not traditional, if you will, and why we may need to look there a little more closely. All this to say that with this kind of approach of looking at phenomics, of brain imaging, of genetics, and bringing them all together, this kind of biologically informed but really data-driven kind of understanding of autism spectrum disorder in boys and girls, I think maybe becoming a little clearer, and that we'll get closer to a point of having an actually biologically based set of biomarkers that tell us who has ASD and who does not, that may be different between boys and girls, linking genetics, neurological health, as well as behavioral metrics to be able to understand autism more carefully. Let me just thank a number of different people who kind of make this possible. I want to thank all of my Autism Center of Excellence colleagues, in particular Kevin Pelfrey, who I've worked with for now almost 15 years on this particular project. I want to thank all of the people who've been at all of the different universities that have participated participated in this, including here at the University of Washington, as well as all of our various funders who've provided funding for a variety of different aspects of this project, whether it was data collection, whether it was the organization of our network, et cetera. There's a number of them, and we really have a lot of students and colleagues who make the team science very fun. And so we're very excited to see this continue into its now third wave of data collection, which, maybe I'll come back in a few years and give you an update on where that's gotten to. And again, if you find yourselves in Charlottesville, Virginia, please come by and visit us. We love to have visitors, and we would love to host our friends from Seattle. So please do come and visit.